I am Bernard Dan, Editor-in-Chief of Developmental Medicine and Child Neurology. It's a great honor and pleasure to have this conversation with Aaron Ciechanover, who won the Nobel Prize in Chemistry for his work on ubiquitination, generally understood as processes that influence the destiny of proteins inside the cell. Aaron, can you tell us about ubiquitin and ubiquitination?
Yes, ubiquitin is a small molecule that is attached covalently to other proteins and tag them, and the tagged protein is now doing something else than it was born to do.
The best known function for ubiquitination seems to be the degradation of proteins, is that right?
So some types of ubiquitination lead to the degradation of the tagged protein, so it's kind of a kiss of death. So ubiquitin in this case kisses the proteins to its death.
And this occurs via the so-called proteasome.
Another function of ubiquitin is to target membrane protein, for example, to the lysosome to be degraded. So it's not degradation by a soluble protease, by the proteasome, but rather degradation by the lysosome.
Yet it isn't all about destruction.
In other cases, modification by ubiquitin or the covalent attachment sends the protein to a subcellular destination that otherwise, without ubiquitin, it would have never gotten there. So this is another function of ubiquitin. So basically, it's a post-translational modification that alters the function of the protein or its destination or its fate. And one of the functions that is the best studied, and that's how we discovered the system, the ubiquitin system, is targeting the tagged protein for degradation by the proteasome, which is a huge, very large proteolytic complex, a destructive complex.
Recent data I read suggests roles of ubiquitination in creating epigenetic marks that regulate gene expression. In particular, I saw that it appears to modify histone proteins, including in neurons, and this may affect synaptic plasticity. On the other hand, in the March 2017 editorial of DMCN on Epigenetics, I also mentioned Angelman syndrome as an example of neurogenetic disorder presumably related to problems with ubiquitination. How do you understand Angelman syndrome?
I think in Angelman syndrome, or it's an imprinted disease, is a ligase, and a ligase is the enzyme that ligates ubiquitin to the target substrate. But since the ligation reaction is highly specific, actually there are close to 1,000 ligases. And the assumption is that some protein has to be ubiquitinated. The protein has not been identified with certainty. There are several candidates by E6AP, and either degraded or targeted to some subcellular destination, the synapse, or somewhere else in the neuron. And because it's not ubiquitinated, it cannot be either destroyed and it accumulates and causes harm, or it's not targeted to the right place, or it's associated with protein that it shouldn't be associated with. Something wrong happens with the lack of this ubiquitination, which leads to this neurodevelopmental syndrome.
Is it known how abnormal ubiquitination can lead to intellectual impairment?
Well, we don't understand much about the mechanism of cognition, but it's clearly that it has to do with the development of the brain. And again, what I said is that, because this enzyme is missing, some downstream target protein is not ubiquitinated, and the ubiquitination is essential for the proper brain development, appropriate movement, development of high functions of the brain. We really don't know. Other substrates of E6AP have been identified, but as you said, they are irrelevant to the brain development. So probably there is another substrate that is yet to be identified that is involved, and it will be very interesting and very important to identify this substrate that is important for the proper development of the brain.
Aaron, how can therapeutic approaches be developed to target disorders of ubiquitination?
For example, you can use the ligase as a bait. You can overexpress it and use it as a bait to hook to it all its substrate, and then you can test the substrate one by one.
Can we gain inspiration from ubiquitin research to design a specific treatment for Angelman syndrome?
Yes, of course, and the methods are there. But again, the model systems will be very, very difficult and very cumbersome. You can take just culture cells, and you can take cultured neurons, for example, even primary neurons. You can either overexpress the protein, the wild-type ligase, or delete it by CRISPR or by silencing it, and then do whole proteome analysis of the cell and see, in the case of deletion, which substrates are coming up. In the case of overexpression, which substrates are coming down. But there is a big caveat here, because you assume a priori that the function of ubiquitination is degradation. But if the function is some other regulatory function, then you may not find it.
So what do you think the next breakthrough will be?
We are still lacking what we call a map of the ubiquitomes. So I'm not talking a map of the degradation ubiquitome, but just a map of the ubiquitome.
Well, quite a bit of work ahead then, delineating the map of the ubiquitin. Many, many thanks indeed, Aaron, for sharing this with us. Take care.